So I think it's the right time to have antibody validation as its own research discipline rather than a simple technical checkbox. We heard a lot of talks, throughout these days. Antibody validation is not trivial. So let's take proteomics as the overarching research disciplines that encompass several subdisciplines such as spatial proteomics, protein quantification and biomarker discovery, binder development, binder validation. Of course, I'm including here antibody validation. And all these subdisciplines are interlinked. With binder validation underpins spatial proteomics to ensure that multiplex assays use reliable reagents while feeding back to binder development. And each of these subdisciplines shares hallmarks of a research field with distinct scientific questions and methodologies and standards, a community, funding, and training. And I believe there's a strong advantage to have antibody validation as a bona fide research discipline. I'm quite convinced it can help bring dedicated funding streams, greater visibility and impact, stronger traction for community-wide efforts, and acceleration toward our shared mission to have validated binders for all human proteins and beyond. So there's an history of key projects that shaped the antibody validation field, well summarized by Rick Kahns and co-authors, their recent review, with many co-authors actually in the room. Most of these initiatives combine large-scale antibody generation programs, antibody validation standards, new data reporting practices. For example, Neuromap pioneered the use of knockout tissues to validate antibodies for brain research, or the five pillars by Uden and al., who suggest antibody validation protocols that are at the basis of most manufacturers today. So I'd like to share some lessons from ICAROS and discuss these milestones for the next years in terms of how can we establish antibody validation as a research discipline. So ICAROS stands for Antibody Characterization through Open Science. It's an open ecosystem. It unites 14 antibody manufacturers and knockout cell line suppliers. They donate their reagents to our lab, where we test them side by side using knockout cell lines in Western blot, in immunoprecipitation, and in immunofluorescence. And now, indeed, we're now extending, thanks to our friends at Leicester, Harvinder and Mike, we're extending to flow cytometry and to immunohistochemistry, thanks to Rihama UB. The funding agencies fund the studies, select the targets for which we're going to test the antibodies for. The antibody characterization data are compiled into a report with one report per protein. These reports are reviewed during our advisory committees. And once we reach our scientific bar, they are quickly uploaded on Zenodo, which is an open science repository. And we gradually publish them at F1000 so that they get peer reviewed and indexed on PubMed. These databases also linked our reports, including CiteAb. This initiative would have never existed without Al Edward, the SGC CEO, who was actually here yesterday. And our protocols are highly inspired by the protocols that Peter McPherson established in his lab. Peter is a well-renowned biochemist and neuroscientist, and he hosts the ICAROS initiative in his lab. Am I forgetting something? So we achieved a major milestone last year with the publication of our consensus industry-endorsed protocols. In yellow, you'll see representative from 11 ICAROS partners that are co-authors. These are protocols for Western blot and IP and IF. And now I'd like to show you what an ICAROS report actually looks like. I selected here GBA1, which is an important Parkinson-related protein. For GBA1, we received 24 antibodies from various suppliers. And you see we follow the same format for each of these antibodies, okay? So you'll see the name of the antibody appearing above each immunoblots. Sometimes there's two stars, sometimes there's one star, sometimes there's no stars. No stars is a polyclonal, one star is a mouse monoclonal from a hybridoma, two stars are recombinant. The antibodies are listed alphabetically. Here's the Western blot screening, obviously. Below each immunoblot, you see the corresponding post-sustained membrane to show that there's equal loading of the wild type and the knockout protein lysate. The wild type cell was selected based on RNA expression. And an ideal GBA1 antibody in that case would provide a specific signal only in the wild type cell, and that antibody wouldn't pick up any protein in the knockout. So for GBA1, most antibodies were recommended in Western blot. However, only 9 were truly selective for the protein. Then we test the antibodies in immunoprecipitation. Just for a lack of time, I'll jump right in the immunofluorescence staining. And I remember Carly saying she did Drosophila work during her training. It's similar with me. And in Drosophila, we can have these mosaic animals that have wild type and knockout tissues on the same Droso. And this is something we're trying to do here. So we played both the wild type and the knockout cell in the same well. And each cell population were pre-labeled with a different fluorescent dye so that we can distinguish the wild type and the knockout. So if we take this one, for example, the wild type cells are outlined in green, the knockout line are outlined in pink. So this antibody here provides specific signal in the wild type cells. There's no signal in the knockout. The signal is comparable to the background. What's interesting here is 5 were recommended in IF. However, in the condition tested, I should always say that, they all failed knockout validation. But we found 4 antibodies that actually showed high specificity that were simply not recommended for this application. So here's our progress so far. So since 2020, we've tested 1,600 antibodies using our consensus protocols, corresponding to roughly 150 protein targets. 18% were removed from the market, and 40% had change in application. But here's a major observation that we made. So high-quality renewable antibodies exist for most proteins simply by screening what's out there, but knockout validation data may diverge from manufacturers' recommendations. So how do we scale knockout-based antibody validation? And I believe is from moving antibody validation as a simple technical checkbox to a bona fide research discipline. So we've discussed already the hallmarks of the research discipline, dedicated scientific questions and training and funding and standards and community. And I'll go through each item. So dedicated scientific questions. How many times do we hear, 80% of antibodies do not work? Or my antibody works in Western blot, it'll never work in immunoprecipitation. What does work actually mean? So here's a list of 5 questions. There's plenty others. Animal vs. non-animal derived binders, which perform best? Does performance in one assay predict another? One of our favorite questions at YCharOS is, how complete is the coverage of human proteins by renewable selective antibodies? And, if we don't have researchers dedicated to address these questions, most, if not all of these questions will remain unanswered. Here's what we do in the lab with our panel of knockout validated antibodies. So here we tested 300 antibodies targeting 33 amyotrophic lateral sclerosis proteins, or ALS. And we used the knockout validated antibodies to profile the expression patterns of these proteins using iPS-derived motor neurons and cortical neurons and dopaminergic neurons, oligodendrocyte, astrocyte, microglia. And what we found is, contrary to the expectation that these proteins would be primarily expressed in the neuron that degenerate in ALS, which are the motor neurons, we found that these proteins actually, there's a high subset of proteins that are heavily expressed in glial cells. And interestingly, this data could not have been predicted through RNA databases. We really needed to look at the protein level. Still within our hallmarks of antibody validation, having a dedicated training. Another great idea from Al Edwards. Let's organize an international course on antibody validation, modeled after the Cold Spring Harbor Laboratory hands-on classes. It could be hosted at McGill or elsewhere at other YCharOS sites. Within a 5 weeks program, the first week could be about classes on antibody validation and antibody epitope recognition, validation. And then the other 4 weeks is bench work. So students, selected scientists would come to the lab, validate antibodies for their protein of interest, following the knockout, the YCharOS protocols. I think it's hard to have students or postdocs to actually read about antibody validation, but when they have the opportunity to come to the lab to validate antibodies for their protein of interest, this is where I think we can have a proper training and have them understand the issue and then go back to the lab and advocate for better validation. And what's I think also cool is that they will test antibodies for their protein of interest and leave with a first author paper at F1000. We hope to have the participation of the industry. There's many ways to participate. Teach a class, have someone do an antibody validation study. We're of course looking for funding. Plan launch summer 2026, and we do have a coordinator, Julia Hu, at McGill. And then the 3 last items, dedicated funding and community and standards. So we're lucky to have the Only Good Antibodies Initiative, OGA, led from Leicester University by Harvinder and Mike. They're working hard on this front. It makes sense, right? Having a strong community and more power would help us to raise more funding and convince journals to raise their standards for antibody validation. So in 2026 or so, I hope we can establish a working group where we can discuss these items, discuss a potential impact of of having antibody validation as a research discipline, perhaps write an opinion paper, and we're poised to launch the first international antibody validation course. So invitation to collaborate. Let's get in touch. Let's start with a working group, opinion paper, and a course. And I would love to thank all the members of the YCharOS team. Riham, you're instrumental to this effort. Thank you so much. The SGC, the OGA, Rick for being supportive, Shade and Reina, our partners. And thank you very much.